Hello, we are going to talk about macromolecules today. So let's take a quick look. A warm up for today, think about what the 4 types of macromolecules are found in living things. Our agenda, we're going to talk about CHNOPS, we're going to do a pattern matching activity, and we're going to discuss macromolecules. Your homework is to continue working on the Chapter 3 notes on biochemistry. And if you're having any difficulty with the concepts, I recommend watching the AP Classroom daily videos to help you to reinforce what we've been doing in class and what we've been talking about in class. So if we think about CHNOPS are the atoms that are the most abundant in living things. So we've got carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So carbon and hydrogen are the 2 most abundant ones. And then we typically see carbon, hydrogen, and oxygen in all of them. But then some of the macromolecules also have nitrogen, phosphorus, and sulfur. And then, of course, there are trace elements that are also found in living things. But CHNOPS are the ones that are the most abundant in living things. So we want to kind of think about each of those different macromolecules that we've got in our bodies. And we want to think about which atoms there are in each one. So you want to take a minute to kind of think about carbon and then which atoms or which molecules, macromolecules, you would find those in. And same thing with hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So if we do that, we take a quick look at that. We're always going to find carbon in all of them. We talked about carbon being the backbone and the basis of all living things. And so we're going to find carbon in all of those macromolecules. And then if we look at hydrogen, also in all of those. So carbon and hydrogen are going to be in all of those macromolecules. Then when we get to nitrogen, we're going to find nitrogen not in everything, but we're going to find it in some things. So we're going to find nitrogen in our proteins, because when we look a little bit later at the structure of a protein and an amino acid, we're going to see that there's nitrogen in there. We're also going to find nitrogen in our nucleic acids, because our nucleic acids are going to have those nitrogenous bases in there as well. So they're going to have nitrogen in both the in the proteins and then also in the nucleic acids. Carbohydrates are simply going to be carbon, hydrogen, and oxygen. And then with the lipids, we're not going to find nitrogen in our lipids. Then when we look at oxygen, we see that oxygen is going to be in all of those macromolecules. So carbon, hydrogen, and oxygen, always in all 4. Then when we get to phosphorus, we see that phosphorus is going to be in our DNA, because if you look at the sugar phosphate backbone of the DNA molecules, you're going to see the phosphorus in there. And then lipids, when you think about the phospholipid bilayer, there's going to be the phosphorus in that. Not all lipids are going to have phosphorus, but those phospholipids are. And then when we look at sulfur, the only one that's going to have sulfur is going to be our proteins, and that's going to depend on whether or not it has that one amino acid that has the sulfur in it. So not all proteins are going to have it, but the ones that do have that particular amino acid is going to have the sulfur. Okay, so keep those in mind as you're thinking about the different macromolecules and you're looking at and trying to identify the different macromolecules. So in class, we're going to do a pattern matching activity where we're going to look at some molecules and we're going to classify them into which group they go into. But we can't do that in the video because I don't have a digital version. So we're going to move on in the video. All right, so looking at macromolecules, there are 4 classes of biological molecules, and so that's what we're looking at with the macromolecules. The Urey-Miller experiment, it was looking at how those organic molecules could have possibly come about on life in the very beginning. So life on Earth started without life. So the planet Earth didn't have life initially. So what the Urey-Miller experiment was trying to do was trying to simulate what the life on Earth possibly could have come about in those early conditions. So they were simulating those early conditions of the planet, and they were looking at those molecules that were in the atmosphere and in the oceans and on the planet, and they were simulating the conditions. So back then it was very hot. You've got lightning happening, you've got molten oceans happening, and then you've got cooling and condensing taking place. And Urey-Miller designed this apparatus that would simulate the early conditions on Earth. And they were trying to figure out, if that was the early conditions on Earth, could those macromolecules form and then form the basis of life on Earth, the basis of those macromolecules. So on the left here, you can see that simulates what the oceans were like. So they were hot and molten. And so we had lots of water vapor going up, and in the atmosphere, we had methane, we had nitrogen, we had hydrogen molecules, ammonia in the atmosphere. So if that water vapor was combining with that atmosphere, was it possible, if that water then cooled and condensed and collected, could those early macromolecules or precursors of macromolecules form? And so they were able to figure out that it was possible to form the precursors for the macromolecules, and that then life could have formed from those precursors. So that was kind of an experiment to see, is it possible that life could form on the planet in the state that it was early on? And so when we look at organic chemistry, what we are studying is we are looking at carbon-based compounds and the reactions and the combination of those molecules into molecules, larger molecules. So organic chemistry is the study of compounds that contain carbon. So carbon dioxide is not considered an organic molecule, but when we combine a whole bunch of carbon-containing molecules together and form macromolecules, now we are looking at organic compounds and organic chemistry. So there are 4 classes again of biological macromolecules. So a macromolecule is simply a really large molecule. So that includes carbohydrates, lipids, proteins, and nucleic acids. And when we are looking at those macromolecules, most of the macromolecules are monomers that get assembled and put together to build polymers. So a polymer is going to be a long molecule that consists of many similar building blocks. And the monomer is the building blocks that you're putting together to make those polymers. And then carbohydrates, proteins, and nucleic acids all fit into that monomer-polymer relationship. The lipids don't, but they still get assembled together with a dehydration synthesis reaction. But they are not considered a polymer, even though they are a large biological molecule. So carbs, proteins, and nucleic acids are all monomer-polymer relationships. So polymers made out of monomers. So when we look at how we make those macromolecules from those monomers or simple subunits, the process is dehydration reaction. So dehydration reaction is bonding together two monomers. And in that process, and the reason why it's called dehydration synthesis is because you are taking out water from that molecule, from that process. And you're creating a water molecule and bonding them together. And then the opposite of that is hydrolysis. So when we take the polymers apart, and break them down into individual monomers, we have to put the water back. And so it's called a hydrolysis reaction. So if we take a closer look at dehydration synthesis and hydrolysis, dehydration, if we look here, we've got a short polymer, and then we want to add this other monomer here on the right. Each of those have either a hydrogen group or an OH group on them. So this short polymer and this monomer, those are molecules that are stable. They're happy, they're full, they've got their electron shells completely full. So they're stable. They really can't bond to anything else unless you break a bond first. And so that's why with dehydration synthesis, we have to remove this hydrogen and we have to remove this OH group so that this molecule here can bond to this molecule here. So this OH and this hydrogen break off and form a water molecule, and then this molecule bonds to this molecule. So number 3 bonds to the new one coming in, and it forms a new covalent bond. The analogy I like to give my students is that imagine that this was you, and you have to go to the park with your little brother or sister. You have to hold hands with your little brother or sister so that they don't get lost. And here is your best friend coming to the park also. They're holding hands with their little brother or sister. But let's say you want to hold hands with your best friend. You're going to have to let go of your little brother or sister's hand. They have to let go of their little brother or sister's hand. Those two kids can go off and play in the park while you hold hands with your best friend. So now you're creating a new bond. You couldn't hold hands before because your hands were busy holding hands with your little brothers and sisters. So if the little brothers and sisters go and play, then you and your best friend can hold hands. So that's that covalent bond that's forming there. Otherwise, those electron shells are completely full and stable. These electron shells are completely full and stable. That covalent bond is not possible unless you break a bond. So you have to break a bond here between this hydrogen and that monomer, and this OH group and this monomer, so that these two can bond together to form a longer chain. And then hydrolysis is the same, but in the opposite direction. If we want to break this bond, you can't just break it because then it's going to be unstable. That electron shell needs to be filled to make that a stable molecule. So what's going to happen is we're going to take the water molecule, we're going to split the water, and then the OH group will make this molecule stable, and this OH group is going to make this molecule stable. So that water splits to then form a hydrogen group here and an OH group onto there. Now they're perfectly stable, and now those monomers can be separated. So hydrolysis requires the water to go in so that it can break the bonds, while the dehydration synthesis needs the water to come out so that those monomers can be linked together. So that's dehydration synthesis and hydrolysis. And all of the macromolecules are going to be assembled using that process, assembled and disassembled using that process. So if we start with carbohydrates first, think about what comes to mind when you think about carbs. So take a minute to just think about what's the first thing that comes to mind when you think about carbs. Most people think of spaghetti or rice, those types of things, bread. Those are your typical carbohydrates. So carbohydrates are going to be those sugars, simple sugars, and polymers of sugars. So they're going to have simple sugars, and those are going to be the building blocks that make up the larger carbohydrates. So monosaccharides are going to be those simple sugars. And so typically when we draw a carbohydrate, we see it as a linear structure. But in water, they're actually going to fold up into a ring structure. So most of the time in biology, we're going to see them as a ring structure. And then those ring structures can be bonded all together. So they're going to form into rings in an aqueous solution. And when we're talking about cells, they're made up of mostly water. So most of the time we're going to see in cells those ring structures. So a monosaccharide is a single sugar, so a single ring structure. If we combine two monosaccharides together, that's going to form a disaccharide. So if we look at this picture here, we've got glucose, two molecules of glucose combining together, dehydration synthesis reaction, and that's going to form a disaccharide called maltose. If we've got glucose and fructose bonding together, dehydration synthesis reaction here, that's going to form sucrose. And sucrose is the sugar that you find typically in table sugar. And then if we combine a whole lot of those simple sugars together, we form a polysaccharide. Poly means many, so lots and lots of molecules all linked together. And there are two types of polysaccharides. We've got storage polysaccharides and we've got structural polysaccharides. Storage polysaccharides are going to be the ones that are storing energy for the cells. There is a storage polysaccharide for plants, which is starch. And then there is a storage polysaccharide for animals, which is glycogen. Both of them are a whole bunch of glucose molecules all linked together, but the glycogen is just linked together in a different arrangement. So it's a little bit more branched than the starch is. So they are both long chains of glucose, but the structure is a little bit different. And then we've got structural polysaccharides, and they are more for forming structures within the cells. The storage ones are storing energy, so an energy storage. So when we're eating our bread or our spaghetti, that is the starch stored in the bread and the spaghetti, and we're getting energy from that. When we look at the structural polysaccharides, animals, at least humans, can't really break down the cellulose from the plant. So we're not going to get energy from that, but it does provide the structure for the cells, the cell walls for the plant cells. So it gives those cells structure. In animals, we also have a structural polysaccharide, and that's called chitin. Typically, you see that in organisms like insects, and that produces the exoskeleton of the insects. But in fungi, it's also the structural polysaccharide that forms the cell walls of the fungi. So similar to cellulose in plants, we're going to find the chitin in insects. We're going to find that chitin in fungi as well. And again, they are very similar to long chains of glucose molecules, but they are arranged a little bit differently. So the structure of them is very similar. Sorry, my battery is running low, so I'm going to run to the other room and plug in. But the structure is going to be similar. You've got glucose molecules bonded together, but they are just arranged slightly differently. So we're going to see a different structure for those molecules. So in this picture, you can see the exoskeleton there of those insects, and they are very strong structural molecules, so strong that they actually use it for surgical thread. So they're very useful and strong molecules. So that is our carbohydrates. Then if we look at lipids, lipids are going to be things that are like our fats and our butter and our oil. So that's typically what we think of when we think of fats. So lipids are also assembled together with those dehydration synthesis reactions, but they're not monomers. They're actually little components that get put together to make a lipid. So the thing about lipids, they are hydrocarbons, and so they are going to be hydrophobic. And so that means that they're not going to dissolve in water. So most lipids are hydrophobic. Phospholipids are a little bit unique because they have a component that is hydrophobic and a component that is hydrophilic, and that is what makes them important for making up our cell membranes. So fats are going to be structured from two molecules. There's a glycerol molecule, and then there are fatty acid molecules, and those fatty acid molecules get attached onto the glycerol molecule with that dehydration synthesis reaction. So glycerol is a 3-carbon molecule, and you're going to add 3 fatty acid chains onto that to make a fat. The linkage, when you're doing that dehydration synthesis reaction, that's called an ester linkage, and it creates a triglyceride. Triglyceride because there are 3 fatty acids attached onto one glycerol molecule. So if we look at this picture here, the fatty acid is this long chain up here in orange, and then the glycerol molecule over here in gray. And so you can see the dehydration synthesis reaction is going to combine this carbon over here onto this carbon over here, making a ester linkage. So we're going to have this bonding together. So that carbon onto that oxygen, which is attached to that carbon. So there is your ester linkage there. You're going to do that 3 times. So you're going to have a dehydration synthesis reaction here, and another one here, and another one here. And you're going to form 3 fatty acids attached onto that glycerol molecule. Fats are high energy bonds, so they are really good for storing energy. So an excellent long-term energy storage molecule. And then in our adipose tissue, we're going to find a lot of fat. So our fat cells have lots of fat molecules in them, so that's storing them. Fats are also, not only are they good energy storage, they're good for insulation. So they're going to insulate our body tissues, and so they help keep us warm, and they also cushion our organs inside of our body. So great for insulation, great for high energy storage. And there are good fats and there are bad fats. So think about saturated and unsaturated fats. You've probably heard about saturated and unsaturated fats. So think about what you know about saturated fats. So a saturated fatty acid is going to have the maximum number of hydrogen bonds possible, and there's going to be no double bonds in there. So we're going to take a look at a picture in just a second. Unsaturated fats have 1 or more double bonds, and so the saturated fats are going to be liquid at room temperature. Sorry, the unsaturated fats are going to be liquid at room temperature, and the saturated fats are going to be solid at room temperature. So the saturated fats typically come from animals, and the unsaturated fats typically come from plants and some fish. So if we look at saturated fats, so you can see here this one on the top, this is a saturated fatty acid. All of these carbons are very, very straight, and every single place you can have a covalent bond with a hydrogen, there are hydrogens there. Well, if you look at the unsaturated fat, in order for this carbon to double bond to another carbon, you would have to remove 2 hydrogens. So it is unsaturated, it's lacking in some hydrogens because that double bond is in there. So it's an unsaturated fatty acid. So if you've got unsaturated fatty acids in your fat, then it is an unsaturated fat. So again, the saturated fats are solid at room temperature. And the reason why they're solid at room temperature is because they are very straight, very linear, which means you can stack a whole bunch of them on top of each other. And because you can stack a whole bunch of them on top of each other, they can pack really closely and tightly together. And so they are saturated fats that are solid. If you look at the unsaturated fat over here, because that double bond is in there, it actually puts a kink in that line, in that chain. And because there's a kink in that chain, it makes those molecules unable to get really close together. So there's more space between those molecules. And because there's more space between those molecules, then they are able to move around a little bit better. And so they are liquid at room temperature. Typically, the saturated fats, again, come from the animals. They are typically the ones that taste really good, but they're bad for you. And then the ones that come from the plants, those are the ones that are healthier for you because they're not clogging your veins and your arteries, and they're not going to lead to cardiovascular disease as much as the saturated fats are. Okay, and then we've got phospholipids. Phospholipids have, instead of 3 fatty acids, they have 2 fatty acids attached onto the glycerol molecule. And instead of that 3rd fatty acid, you've got a phosphate group attached onto the glycerol molecule. And that phosphate group has a charge. And that's why that charge in the phosphate group makes that end of the molecule charged, which is then able to be dissolved in and have an affinity for the water. So you're going to have a hydrophobic end and a hydrophilic end when we look at phospholipids. So here's your diagram. Here is your glycerol molecule up top. This time it's kind of going horizontal instead of vertical, but your fatty acids are these 2 orange chains coming down. This one is a saturated fatty acid chain and an unsaturated fatty acid chain, so it's putting a kink in it. And then up here is a phosphate group, and you can see negative charges on the oxygen, which makes this negatively charged. So this phosphate group is hydrophilic, and then these hydrophobic tails are not going to like the water. And that's what gives the cell membrane its properties. Cell membranes are made up of a phospholipid bilayer. So here's another diagram. It's now turn to the side, but you can see your glycerol molecule here, 2 fatty acid chains, and then a phosphate group over here. Again, you can see those charges on that phosphate group. And when we get to cell membranes, we'll come back to phospholipids again because they make up an integral part of the cell membrane. So here you can see that phospholipid bilayer. The hydrophilic end is going to face where the water is. So we've got water inside the cell, we've got water outside the cell, and then the hydrophobic tails are going to hang out with each other facing inside of that bilayer. Again, we'll come back to that. Another type of molecule in the lipid category is a steroid. So steroids are going to be lipids. They are characterized by a 4-ring structure. So anytime you see 4 rings linked together in that arrangement, that tells you that it's a steroid. Cholesterol is an example of a steroid. It plays an important role in cell membranes. So when we get to our membrane unit, we'll talk more about cholesterol. But there are lots of steroids and lots of hormones that are steroids that play an important role in the body. But they are all characterized by these 4-ring structures. So those are our lipids. So we've got fats, phospholipids, and steroids. Waxes would also fit into that category. So those are going to be hydrophobic molecules. Then moving on to proteins. When we think about proteins, we think about steak, we think about fish, we think about chicken. Lots and lots of eggs would definitely be something that comes to mind when we think of proteins. Proteins actually make up most of the dry mass of the cells. So more than 50% of the dry mass, so if you take out the water, what's left is going to be mostly proteins. And proteins do all of the work in the cells. They're doing all the jobs in the cells. They're getting the work done. And there's different functions of those proteins. So we've got lots and lots of different functions in our proteins that are in our cells. So we've got some proteins that form and function as enzymes, so they speed up chemical reactions. We've got ones that work in our immune system to help fight off infection. We've got storage proteins like in our eggs. We've got transport proteins that help move things into and out of the cells. And we'll talk about a lot of these when we get to later units. Some of them are hormonal proteins, so think of insulin regulating your blood sugar level. We've got ones that work in sending signals, so like in our nervous system. We've got contractile and motor proteins like our muscles. We've got structural proteins that kind of help to form the structure in our cells. Our hair is made up of structural proteins like keratin. So lots and lots of different functions in our body. Again, enzymes are those types of proteins that speed up chemical reactions. So they work as catalysts, and they can be working to either put molecules together or take molecules apart or just rearrange one molecule into another molecule. So lots of different even enzyme functions. A lot of chemical reactions require those enzymes to facilitate those chemical reactions. And we will see that as we move through the year. We'll talk about lots and lots of enzymes as we go. Proteins are, again, polymers. And so they are going to be made up of amino acids. So the polypeptide, a polypeptide, is a long chain of a whole bunch of amino acids all bonded together. There are 20 different amino acids, and the sequence of those is determined by your DNA, which we will talk about in a later unit as well. But the protein, a protein, is a biologically functional molecule that consists of one or more polypeptides. So when we look at those definitions, a polypeptide is going to be the long chain of amino acids, which then usually does fold up into a protein. The protein is what is functioning in the body. So after the polypeptide chain has been made, it typically will fold up into a unique shape, and that shape determines its function. So it is called a protein once it is functioning and has folded up. But when it comes off of the ribosome, it is called a polypeptide. Some proteins are made up of multiple polypeptides put together. And so the protein, again, is going to be that final overall shape of that molecule. So the polypeptide is the long chain of amino acids, and then the protein is its overall shape. So if we look at the monomer for proteins, we are looking at amino acids. All amino acids have a basic shape, and so we can look at that basic shape over here. So we've got an amino group attached onto a central carbon, and then we've got a carboxyl group also attached onto that central carbon. And then attached onto that central carbon, we've got a hydrogen down here. And then we've got an R group up here. What makes one amino acid different from another is what side chain you have as your R group. Everything else down here is going to be exactly the same in every single amino acid. So every amino acid has that central carbon with an amino group and a carboxyl group attached onto it. And then that central carbon has an R group. And again, that R group is what differentiates one amino acid from another. Those R groups are going to have different characteristics. Some of those R groups are going to have nonpolar side chains. And so if they are nonpolar, then they are going to be hydrophobic. And that's going to cause the protein to fold in a unique way so that those side groups are moving away from where there is water. So usually they kind of get tucked on the inside of the shape of the protein. And then sometimes the side chains are going to be hydrophilic. And so there are a set of amino acids that are hydrophilic. And then we've also got some that have a complete charge. So they could be negatively charged or positively charged, but they have an electrical charge. They are also going to be hydrophilic because of that charge is attracted to the water. So those R groups, those side chains, are going to kind of determine how the protein folds up. So some of them, again, are nonpolar, and so they don't have a charge. Some of them are polar, and so they have a partial charge, or they could be electrically charged and have a full positive or negative charge. And so those are then those amino acids, when they're going through the ribosome and they are bonding together, they're going to bond together with that dehydration synthesis reaction again. And the bond that they're going to form is called a peptide bond. You're going to have sides to those molecules as well. So you're going to have one end that is closest to the carboxyl end, and that's called the C-terminus end. And the other end is going to be the side that is close to the amino acid or the amino end. And so that's the N-terminus. So we go back to this picture here. And so if you imagine putting a whole bunch of those side by side, you've got an amino group on the left and you've got a carboxyl group on the right. So there's going to be an N end and a C end to that chain once they are all put together. Let's see if we can find a picture. Yeah, here's a picture of them combining together. So this is going to be your N-terminus over here because that's closest to the nitrogen end. And then you're putting it together with that dehydration synthesis reaction. And then this end is going to be your C-terminus end because that's the end that's closest to that carbon end. So you're always going to have, once they're all bonded together, you're going to have a backbone that goes nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. And so that's going to keep repeating over and over as you go down that amino acid chain. Once you've got that amino acid chain formed, then it's going to fold up and it's going to have interactions because it's in an aqueous solution, it's in a water environment, that's going to cause that protein to fold up. And it's going to form into a three-dimensional shape depending upon the amino acid sequence. And that's going to give it its unique function. So in biology, we always say that structure equals function. And what that means is the shape of the folded up protein determines how it works in the cell. So its structure will determine its function, how it works in the cell. So for example, here we've got an antibody. And that is a protein that has a unique shape that will fit onto a virus or a pathogen because it fits a unique shape that matches the shape of that pathogen so that it can attach onto it and it can disable that particular pathogen so it doesn't get us sick. So again, structure equals function. And how the protein folds up into its unique structure, we need to look at protein folding and the 4 levels of protein structure. So there is the primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure of the protein is going to be its unique amino acid sequence. So the order that it comes off, the order of amino acids that it comes off of the ribosome, that is its primary structure. Then it starts to fold up. As it's coming off the ribosome, it's going to start to fold up. The secondary structure is going to be, if we look at the backbone of an amino acid, let's go back to that here. Let's go back right here. So if we look at this purple area, in that purple area, we can see a lot of hydrogens sticking off of there. And then we've got some oxygens in there as well, and we've got some nitrogens in there. So anytime we've got interaction in that backbone, hydrogen bonding occurring in that backbone, that is your secondary structure. So secondary structure is going to be when you've got hydrogen bonding in the backbone. And typically with secondary structure, we find it either forms into coil-like structures, or it could fold back and forth upon itself, and that forms what we call a beta-pleated sheet. So if we look at some pictures, here's your primary structure again, the sequence of amino acids. And then here is our secondary structure. So it's either going to form these helical shapes called an alpha helix, or it could fold back and forth upon itself, and that's called a beta-pleated sheet. So there's going to be areas in a protein when it's all folded up that are hydrogen bonds that are forming these sheets or layering structures of the sequence. And so those are called your secondary structures. And that's because of hydrogen bonding in that backbone. And then if we go back to our definitions here, our tertiary structure is going to be anytime we're looking at interactions in the R groups of that amino acid sequence. So again, the R groups are these things that are now sticking up, and they're the ones that are different. Again, some of them are hydrophilic, some of them are hydrophobic, some of them are charged. So all of those interactions between those R groups and of course, the aqueous environment is going to cause the protein to fold up into its unique shape. So the tertiary structure is the protein folding up, and those R groups are interacting with each other. Typically, those things that are hydrophobic are going to get tucked on the inside of the protein away from the water as much as possible. And then things that are charged are going to be more on the outside. So if it's a polar R group, it's going to kind of be more on the outside of the molecule. We can have things like hydrogen bonds holding strands together. We can have covalent bonds, disulfide bridges between those R group interactions. If we've got charged R groups, they can have ionic bonds forming in those R groups. Anytime you've got any interaction between any of those R groups, whether it is a hydrogen bond or a covalent bond or an ionic bond or just hydrophobic interactions, those are all your tertiary structure of your protein. And then again, going back to our definitions here, the quaternary structure is if you have multiple polypeptide chains, then you're going to have R group interactions between those polypeptide chains, forming the overall structure of the molecule, and that's the quaternary structure. So not every single protein has a quaternary structure, but if they are made up of multiple polypeptide chains, like this hemoglobin here is made up of 4 polypeptide chains, all to form that one hemoglobin protein, that is going to be held together again by R group interactions between those polypeptide chains, making the overall structure of that particular protein. And here's just a little GIF that's showing you protein folding and those R group interactions folding up that protein into its overall shape. And here's another animation showing that protein folding up into its final overall shape. So R group interactions forming the shape of the protein. And then our final macromolecule is the nucleic acids. And when we think of nucleic acids, what comes to mind? Most people think of genes or DNA or genetic material, but the nucleic acids are all of those. It's typically our DNA and our RNA. So when we look at nucleic acids, that's going to be what stores and transmits our hereditary information. And we do have DNA and we've got RNA that all fit into that category. Nucleic acids are made up of those nucleotide monomers. And then DNA is going to provide its instructions for replication. And then the DNA is going to direct the process of making RNA molecules. So all of the hereditary information is stored in that DNA molecule. So all the instructions for life are in that DNA molecule. So when it's time to read that DNA molecule, it has the instructions for making RNA and instructions then in the end for making proteins as well. And we will talk more about protein synthesis a little bit later in the year. So this is just a quick overview of nucleic acids. But again, it is a monomer-polymer relationship. So it is a polynucleotide because it's made up of a whole bunch of monomers called nucleotides. And a nucleotide has 3 components to it. It has a sugar, and attached onto that sugar is a phosphate group and a nitrogenous base group. And then they kind of get all stacked together and form into a long chain of nucleotides that then all combine together. DNA is going to be double-stranded, so it's actually 2 strands hydrogen bonded together. And then RNA is going to be a single-stranded molecule. And then if we look closely at those nitrogenous bases, we have 2 families of nitrogenous bases. We have pyrimidines and we have purines as nitrogenous bases. And then we've got different sugars in our DNA versus in our RNA. So first, if we look at the pyrimidines versus purines, pyrimidines are going to be these single-ringed structures, and that's our cytosine and our thymine in our DNA. And then in uracil, we're going to have cytosine and uracil as our pyrimidines. And then we have our purines are going to be these double-ringed bases. And so we've got adenine and guanine as our purines. When we look at the base pair rule, we're going to have a pyrimidine bonded to a purine. And then we've got our 2 different kinds of sugars. DNA is going to have deoxyribose sugar, and RNA is going to have ribose sugar. So if we look at the structure of ribose versus deoxyribose, they are the same, except for if we look at this carbon here, it has an OH group in the ribose category, but in the deoxyribose, it is missing an oxygen, hence the name deoxyribose. So deoxy means without an oxygen. So it's missing that OH group there, and it just has a hydrogen there. So very, very similar, just missing an oxygen in that deoxyribose. And then when you look at the nucleotide for it, you're going to have cytosine, thymine, adenine, and guanine nucleotides in the DNA, and then you're going to have cytosine, uracil, adenine, and guanine for your ribose RNA nucleotides. And then when you put them all together, when you are bonding those nucleotides together, it forms a phosphodiester bond between them. Again, it forms that bond through that dehydration synthesis reaction. So again, the DNA molecule is going to be a double helix, and the RNA is a single helical structure. And the DNA molecule actually is going to, again, have two strands, but they're going to be running in opposite directions, and we call that anti-parallel structures. So the DNA molecule runs anti-parallel to each other. And one DNA molecule can have multiple genes, but again, we're going to get into all of this a little bit later in the year. So the base pair rule for DNA, adenine always bonds to thymine, and guanine always bonds to cytosine. So that's called complementary base pairing, or the base pair rule. When we're looking at RNA, adenine is always going to bond with uracil, and guanine is always going to bond to cytosine. So we look at that structure. Here is your DNA molecule, double-stranded, double helix. And the RNA is going to be single-stranded, but it can fold up and it can have parts that do bond together to each other. So the tRNA molecule actually does kind of fold up into a similar T-shaped structure. And then when we look at the DNA nucleotide, or the RNA nucleotide, we can actually number the carbon. So here is our sugar. And if we look at that shape, each corner here is a carbon molecule. And if we number the carbons in our DNA molecule, this is carbon number 1, then this is carbon number 2. So we're going around the molecule in a clockwise fashion. So again, you're starting at the oxygen and then going to the right, carbon number 1, carbon number 2, carbon number 3, carbon number 4, and sticking up from carbon number 4 is carbon number 5. The nitrogenous base group is going to be attached onto carbon number 1. And then the phosphate group is attached onto carbon number 5. When a nucleotide is bonded to another nucleotide, it comes in actually down here from the bottom, and the phosphate group will bond on to carbon number 3 to add a new nucleotide. And so when we look at the directionality of the DNA molecule, there is a 3' end and a 5' end, and there is a 3' end on this one and a 5' end on that one. But they are anti-parallel. So the left side runs 5' to 3' if you're going from top down. And then on the other side, again, going from top down, this end goes 3' to 5'. And how you can tell which end is which, if you look at the bottom down here, this bottom is closest to carbon number 3. And that's what makes that the 3' end. If you look at the top up here, that top is closest to that phosphate group, which is attached to carbon number 5. So that makes that end the 5' end. So this molecule runs 5' to 3'. On the other side, it's upside down. So this end here up top is closest to carbon number 3. So that's your 3' end. And then down here, this is closest to carbon number 5. So that is your 5' end there. So it's anti-parallel. One strand runs in 5' to 3'. The other strand runs opposite, 3' to 5'. You can also just look at the picture and you can see on this side, the oxygens are all facing up and these oxygens are all facing down. That's because the DNA molecule is anti-parallel. And then when we look at how those molecules bond together, we have adenine bonding to thymine, and we've talked about that base pair rule. And the base pair rule works the way it does because those hydrogen bonds have to actually match up. So adenine and thymine are going to form 2 hydrogen bonds, and guanine and cytosine will match up. The hydrogen bonds will form, and it forms 3 hydrogen bonds. We'll get into a little bit more about that hydrogen bonding a little bit later, but for now, just know that adenine and thymine form 2, and guanine and cytosine form 3 hydrogen bonds. And then if you remember back to 9th grade bio, and you saw this picture of Rosalind Franklin's X-ray crystallography picture, from her picture, Watson and Crick were able to figure out how wide the DNA molecule was. And from that width, they could figure out that you had to have a purine bonding to a pyrimidine to fit into that width. And that again led them to figure out that base pair rule, that plus Chargaff's principle or Chargaff's rule that he discovered in nature, that you always have equal amounts of A's and T's and equal amounts of C's and G's. And again, we'll get into that even more when we get to our DNA unit. And then this is just showing you how those polymers are assembled. So here you've got the DNA polymerase enzyme that is adding the new nucleotides. And when a new nucleotide comes in, it actually is a nucleoside triphosphate that is being used to put nucleotides on. And again, that phosphate group is going to attach onto the 3' carbon of the previous molecule that's already on the building strand. And so the DNA polymerase will actually remove 2 phosphate groups and then covalently bond that nucleotide on to that growing strand. And DNA polymerase can only do it in that way, so you always have the new strand being made 5' to 3'. But again, we'll get into that a little bit more as we go. And so that kind of wraps up our overview of the macromolecules. And please let me know if you have any questions. If I can stop the video. Okay. All right. Goodbye.